This is a presentation on the chemistry of drugs and medicine. What is a drug and/or medicine? A drug or medicine is any chemical substance that, when introduced into the body, alters how the body functions. Medicine and drugs. A medicine is a drug that is used intentionally for beneficial effects, such as preventing, diagnosing, or treating disease, relieving symptoms, or improving health. Though this doesn't mean that it can't be harmful, the side effects can happen, and they do. While a drug can refer to any substance that changes physical and mental function, its effects can be both beneficial, meaning therapeutic, and or harmful. This can be caused by either the dose administered, how it is used, or by the individual taking it. Key points about effects. Therapeutic effects is the intended beneficial effect, such as reducing pain or fighting infection, like previously mentioned. Side effects. Unintended but sometimes mild effects. Some medicines' side effects can sometimes become the therapeutic effect, as seen with the drug sildenafil. It was originally developed to treat high blood pressure and heart problems, but during testing the side effects became the new therapeutic effect, as it could be used to treat erectile dysfunction and became later known as Viagra. Adverse effects. Harmful or dangerous reactions. Dose-dependent effects. Small amounts may help, but larger doses may cause harm. Placebo. A placebo is a substance or treatment that has no active medicinal effect, but is given to the patient as if it was a real medicine. A placebo is often used in clinical trials to be compared against a real medicine. It helps researchers to determine whether the actual drug works better than the psychological expectations. The placebo effect is where sometimes a patient can feel better after taking the placebo because they believe that they are in fact receiving the real treatment. Sometimes, something as simple as a sugar pill, saline injection, or a tablet with no active ingredient can be placebo in question. Therapeutic window. The therapeutic window, or beneficial window, is the range of the drug doses that produces the desired therapeutic effect without causing harmful side effects. The key point is that it lies between the minimum effective dose, MED, the smallest dose that produces a beneficial effect, and the minimum toxic dose, MTD, the lowest dose that begins to cause harmful effects. Tolerance. Tolerance in the medical field is when the body becomes less responsive to a drug after repeated use, leading towards larger doses being needed to produce the previous therapeutic effect, the beneficial effect. This can develop after regular or prolonged use of the drug, making the original dose to be ineffective over time. For example, in opioids, this can lead to high dependence and development of addiction. This can be seen by multiple people. More known was with Elvis Presley. He was taking painkillers and he eventually died from them. Tolerance can occur when the body adapts to the drug. The receptors in the body become less sensitive. The drug may be broken down faster by the body. Side effects. Side effects in the medical field are unintended or secondary effects that occur in addition to the desired therapeutic effect of a drug. They can happen because the medicines can affect multiple systems in the body, not just the target area. Side effects can be mild, moderate, or severe, sometimes even debilitating, that can lead to even death. Not everyone who takes medicine will experience side effects, and sometimes not the same ones, as it can affect everyone differently. Similarities and differences in the structures of related drug molecules. This drug molecule. Sometimes related to drug molecules are drugs that belong to the same chemical family, and therefore have similar molecular structures, but with small differences in certain groups and or atoms. Similarities in the structures of related drug molecules. Related drug molecules often have the same basic molecular framework or backbone, similar functional groups, similar overall shape and size, similar chemical properties and mechanisms of action. Because of these similarities, they often act on the same biological target in the body. Differences in the structure of regulated drug molecules. The differences that are of small changes in functional groups such as these here. Different side chains attached to the main structure. Slight differences in molecular arrangement or length. These small differences can change the drug's strength or potency, duration of the action that they are subjected, side effects, solubility or absorption in the body. Functional group modifications. Functional group modifications is the process of changing, adding, or replacing a functional group in the drug molecule to alter its properties. A functional group is a specific group of atoms in a molecule that determines how the molecule behaves chemically. By modifying these groups, scientists can change how the drug works in the body. They are modified to improve effectiveness, drug delivery, and safety. How functional group modifications can increase the effectiveness in drugs. Functional group modifications can improve the effectiveness of drugs by changing how the drug interacts with its biological target, including how it behaves in the body. By increasing the binding to a target, adding or changing functional groups can help form stronger interactions to receptors or enzymes. This creates better binding, leading to more potent and effective application of the drugs. Better selectivity. Many drugs can potentially interact with multiple receptors or enzymes in the body. This can reduce the effectiveness of the drug in question and may lead to unwanted effects. Functional group modifications can change shape, polarity, or change distribution of the molecule. These changes can make the drug fit better into a specific binding site, like a blocking key. Improved selectivity means the drug acts mainly on the intended target, less interaction with other biological molecules, and more effective treatment with fewer unwanted effects. Improved solubility. Solubility is how a substance can dissolve into another substance to form a solution, hence the word solubility. For a drug to be effective, it must reach the target in the body. Functional groups can influence how well a drug dissolves and its path through biological systems. Adding polar functional groups in these examples can increase water solubility, helping the drugs dissolve in the blood and bodily fluids. Adding non-polar groups, for example hydrocarbon chains, can increase lipid solubility, lipid meaning fat, which helps the drug pass through cell membranes. Improving solubility and absorption ensures that the more of the drug reaches the bloodstream and target tissues, increasing the effectiveness. Longer duration of action. Functional group changes can also affect how quickly the body metabolizes and removes the drug. If the drug stays in the body longer, it can maintain its therapeutic effects, beneficial, for longer periods, leading to fewer doses needed. Some modifications are that they protect the molecule from rapid breakdown by enzymes, make the drug more chemically stable, allow the drug to remain active in the bloodstream for longer. How functional group modifications can improve the delivery of drugs. Improving stability in the drugs. For a drug to be delivered effectively, it must dissolve in the bodily fluids such as blood, which is mostly water. If a drug does not dissolve well, only a small amount will be absorbed. Functional groups affect the polarity of a molecule. Polar functions such as hydroxyl, carboxyl, and amine can form hydrogen bonds with water molecules. This increases water solubility, allowing the drug to dissolve more easily in the bloodstream. Non-polar drug groups, such as hydrocarbon chains, increase lipid solubility, which can help the drug dissolve in fatty environments in the body. By adjusting the balance between water solubility and lipid solubility, scientists ensure that the drug can travel in the blood but also pass in tissues when needed. Helping the drug cross cell membranes. Cell membranes are made of mainly a phospholipid bilayer, which is a double layer of phospholipid molecules that forms the main structure of the cell membrane, which has a fat-like interior. Drugs must pass through this barrier to enter cells or internally absorbed by the digestive system. Functional group modifications can be increasing the lipid solubility, allowing the drug to diffuse through the membrane more easily, adjust the size and polarity of the molecule so it fits better through biological barriers, and for example, adding non-polar groups can make the molecule more compatible with the fatty membrane interior. This can improve oral absorption, meaning more of the drug moves from the stomach or intestines into the bloodstream, improving stability in the body. Some drugs can break down too quickly due to the stomach acid, which is hydrochloric acid, which is a very powerful acid that exists. Enzymes in the liver or other metabolic processes. If this happens, the drug may be destroyed before reaching the desired target site, rendering the drug useless. A protective coating around the drug can lessen the odds of this happening. Functional group modifications can make the molecule chemically more stable, reduce how quickly enzymes can metabolize the drug, protect certain parts of the drug from hydrolysis and oxidation. For example, replacing a reactive functional group with a less reactive one can slow down degradation. This allows more of the drug to remain intact during transport through the body, improving delivery to the target. Targeted delivery to specific areas. Functional group changes can help drugs interact with specific targets, with specific transport proteins, tissues, or receptors in the body. This can guide the drug toward cells or organs. For example, adding certain groups may allow the drug to bind to transport molecules in the blood. Some functional groups can help drugs accumulate in certain tissues where the target receptors are already located. This improves delivery by ensuring that more of the drug reaches the intended site of action, while less affects other parts of the body. How functional group modification can improve the safety of the drugs. One minute, so have a drink. Reducing toxicity. Some functional groups in the molecule can be highly reactive or can form toxic metabolites. While the drug is broken down in and by the body, this can damage tissues or organs, usually the kidney or liver. By modifying or replacing these groups, chemists have been able to make the molecules less chemically reactive and therefore less likely to interact with biological molecules that are important, like that of proteins or DNA. For example, a reactive functional group may be replaced with a more stable group. This reduces the chance of unwanted chemical reactions occurring in the body. As a result, the drug becomes less toxic while still maintaining its therapeutic effect. Increasing selectivity. A drug enzyme can sometimes bind to receptors or enzymes in the body. When this happens, it may produce unwanted biological effects which appear as side effects. Functional group modifications can change the shape, size, polarity, and change distribution of the drug molecule. These changes allow the drug to fit more precisely into the binding site of the intended target. This improves selectivity, meaning the drug mainly interacts with the correct receptor or enzyme, which is less likely to interact with other biological targets. Greater selectivity reduces off-target effects, making the drug safer for the patients. Controlling drug breakdown through metabolism. The body removes drugs mainly through metabolism by enzymes, especially in the liver. Functional groups strongly influence how easily these enzymes can recognize and break down the drug. If a drug is metabolized too quickly, it may accumulate in the body and reach toxic levels. If it is breaking down too quickly, very high doses might be needed, which also increases risk. By modifying functional groups, scientists can adjust how easily enzymes attack or modify the molecule, control the rate at which the drug is broken down. This helps maintain drug concentrations in the bloodstream and prevents harmful buildup. Reducing the harmful side effects. Functional group modifications can influence how drug moves through the body, its distribution, which tissues it reaches. For example, modifying functional groups can change the drug's solubility and polarity, affecting how easily it enters certain tissues. This can help limit the drug's presence in non-target organs where it might cause damage. By directing this drug mainly towards the target area, scientists reduce the likelihood of unwanted psychological and physiological effects, improving the overall safety of the treatment. designating compounds as cis or trans isomers. What are cis and trans isomers? Some molecules have double bonded or a ring that stops parts of the molecule from rotating. Because of this, the groups attached to the molecule can be arranged in two different ways. Cis, the important groups that are on the same side of the molecule, which is this. Cis isomer is a type of geometrical isomer where similar or identical groups are positioned on the same side of the double bond or ring structure. The word cis means on the same side, which describes how the groups are arranged in the molecule. This arrangement occurs between the molecule because the molecule cannot freely rotate in a carbon-carbon double bond. The presence of this bond locks the atoms in place. This means that once the groups are on the same side, they stay in that position unless enough energy is applied to break the bond. This is a cis isomer and a trans isomer, slightly different. For example, it is but 2-ene. The cis isomer has methyl groups on the same side of the double bond. This makes the molecule slightly bent or less symmetrical compared to the trans isomer. Because the groups are on the same side, because the groups on the same side, their effects do not cancel each other out. This usually makes cis isomers more polar, meaning that there is an uneven distribution of charge across the molecule. As a result, cis isomers have stronger intermolecular forces and may have higher boiling points than their trans counterparts. In drug molecules, this difference in shape is very important. The body relies on very specific interactions between molecules, often described as a lock and key fit. A cis isomer may have the correct shape and fit precisely into a receptor to bind to a biological target, making it effective as a drug. Trans isomers. A trans isomer is a type of geometrical isomer where similar or identical groups are positioned on opposite sides of a double bond or a ring structure. The word trans means across, which helps describe how the groups are arranged. This happens because the molecule cannot freely rotate in a carbon-carbon double bond, C=C. Because of the bond, called a bond, locks the atoms in place. Because of this, once the groups are arranged on opposite sides, they stay that way unless a large amount of energy is used to break the bond. The difference of geometrical isomerism to the functioning of drug molecules. The significance of geometrical isomerism in the drug molecule is that it affects how well a drug works in the body, even though the isomers have the same atoms and formula. Geometrical isomers, such as cis and trans, have different shapes, and in biology, shape is very important. Drug molecules must fit into specific targets in the body, such as receptors and enzymes. This is often described as a lock and key mechanism, where only the correctly shaped molecule will fit properly. Because cis and trans isomers have different shapes, they behave very differently in the body. One isomer may fit perfectly into the target and produce the desired therapeutic effect, while the other may not fit well and therefore be less effective or completely inactive. A good example is cisplatin. If a cis isomer works as a chemotherapy drug, because its shape allows it to bind to DNA in cancer cells and stop them from dividing, the trans isomer cannot bind in the same way, so it does not work effectively as a cancer treatment. Chiral molecules. A chiral molecule is a molecule that cannot be superimposed in its mirror image, as it forms two versions that are like the left and right hand in a mirror image. These mirror image forms are called optical isomers or ionomers. To identify a chiral molecule, one must look to see if it contains a carbon atom, asymmetric. This carbon atom is bonded to four different atoms or groups. In any two groups that are the same as the molecule is not chiral. The key features are that are that chiral molecules all have the same molecular formula and bonding, different 3D arrangement and spacing, and cannot be placed exactly on top of each other. This is an example. Optical isomerism. Optical isomerism influences how drugs work because two isomers, enantiomers, have different 3D shapes but the same atoms, while the body is a very specific shape. The different effects they may have in the body is that of one optical isomers may produce the desired therapeutic effect, be less effective, may be inactive or even harmful to the body. This means that the same drug may have very different outcomes depending on the isomer used. Lock and key fit with receptors. Drugs work by fitting into receptors or enzymes in the body, like a key fitting into a lock, hence the name. One isomer may fit perfectly and activate the receptors, or another would not fit properly, leading to lower potency. Because of how well they bind, one isomer may be more potent than the other with a lower dose, and the other can be less effective even with higher dosage. Side effects and safety. The less suitable isomer may have interactions with other parts of the body, leading to unwanted side effects or toxicity. There is a reason drug safety is very important, very closely linked to optical isomers. This is an example of the lock and key model. Here is an enzyme. You've got the binding of the substrate. You've got substrate A, substrate B, and you've got the product. Drug detection. Drugs can be detected in a multitude of ways through analytical techniques. These identify and measure substances in samples such as blood, urine, or tablets. Methods such as these work by separating, identifying, or measuring chemicals based on their properties, though they are not inflammable and may not always detect them. And people in sports trying to find a way to get around them. Chromatography. Chromatography is a separation technique. It is used to separate the different substances in a mixture. A sample is placed in a mobile phase, such as liquid or gas. As it moves through a stationary phase, the different substances travel at different speeds, leading to the separation, making them split apart. Examples of this are thin layer chromatography and gas chromatography. Mass spectrometry, or MS. Mass spectrometry is used to identify molecules based on their mass. The sample is ionized, meaning charged. These ions are then separated based on their mass to charge ratio, M/Z. A mass spectrum is then produced. This gives a unique pattern or fingerprint for each group, allowing accurate identification. This is an example of a mass spectrometry showing all what happens. Colorimetry. It measures the connection of a drug in a solution. A region is placed to react with the drug to produce a color change. The intensity of the color is measured. The deeper the color, the higher the concentration of the drug in question. These are 4 examples that can be used. CRLS, the Checker HC Hanimer, the Exilon Analytics STD colorimeter, and the laboratory colorimeter WPA CO7000 biochrom. Combined techniques are more effective. Combined techniques is simply where 2 or more of the previous techniques are used in conjunction with each other. This is considered the most accurate and reliable method used for drug detection. Identifying new drug molecules. The way to identify new drug molecules is through numerous ways, such as screening and natural resources, chemical synthesis and modification, computer-aided drug design, which is molecular modeling, high throughput screening using knowledge of biological pathways. How computers are used in drug design. Computers are used extensively in modern drug design by helping scientists model, predict, and test drug molecules quickly and effectively before they are made in the labs. This saves time and money. Modeling the target. Scientists use computers to create 3D models of biological targets such as proteins or enzymes. The exact shape of the active site can be determined through this process. This allows scientists to see where a drug molecule can be binded. This is a very important distinction to be made, as these drugs must fit perfectly into their targets. Designing drug molecules. Once the target is known, computers can be used to design molecules that will fit better into it. Different molecular structures are created virtually, or functional group modification groups can be added or changed on the screen. The shape and size of the molecule can be used to adjust the screen. As mentioned before, they can save a lot of time and money. Predicting how well a drug will bind. Computers can simulate how well and strongly a drug will interact with its target. This is called molecular docking. It shows how the drug fits into the binding site and estimates the strength of the interactions that take place, reducing time and cost. Using computers means fewer chemicals need to be made and tested. Through this, less time is spent on the previously done trial and error that used to be done in laboratories. This was the most common way of doing things before. Testing properties virtually improving existing drugs. Testing properties virtually can be done with computers to predict the solubility, stability, and toxicity the drugs may have. Improving existing drugs can be done with computers to modify previous drug structures or predict how the changes will affect performance. Thank you for listening. These are my references, all the ones that I've used. Thank you for hearing me.